Reproduction in organisms. Children able to know about definition of reproduction, mode of reproduction, and different types of asexual reproduction. Reproduction is the biological process by which new individual organisms are produced from their parents. In all living organisms, reproduction is the necessary life process for continuation of their life. There are two forms of reproduction: asexual reproduction, sexual reproduction. Asexual reproduction. First of all, we will look at sexual reproduction. In sexual reproduction, male and female organisms produces male gametes and female gametes. These both gametes fusions together. That is called fertilization. Because of fertilization, zygote is forms. Further, zygote develops into new organism. But whereas in asexual reproduction, there is no male and female organisms are involved. Only a single organism produces new organisms. This is called asexual reproduction. Fission, budding, fragmentation, regeneration, vegetative propagation, spore formation, some of the asexual reproduction. Fission. This mode of asexual reproduction we can see in unisexual organisms such as amoeba, paramecium and bacteria. Parent cell divides into two daughter cells or many daughter cells. Budding. Parent cell produces a small bud-like structure. Gradually, this bud increases in size and finally it separates from the parent cell and survives as a new organism. Fragmentation. Organisms break up into smaller pieces. Upon maturation, each piece grows into an independent organism. Regeneration. When cut into pieces, each fragment of the some organisms grows into an individual organisms. Vegetative propagation. Of reproduction in plants by which vegetative parts, roots, stem, leaves develops into new plants under appropriate conditions such as runners, rhizome, sucker, tuber, offsets, bulb are giving rise to new plants. Some are examples of vegetative propagation. Spore formation. The common bread mold consists of fine thread-like projections, thin knob-like structures called sporangia. Each sporangium contains spores. After maturing the sporangium, the spores are released from the sporangium. Artificial vegetative propagation. Vegetative propagation is an asexual method of reproduction that occurs in its leaves, roots and stem. Artificial vegetative propagation. A method of vegetative propagation is carried out by humans in the fields and laboratories. The most common type of artificial vegetative reproduction techniques includes cutting, layering and grafting. Cutting. In this, a portion of a plant is cut and planted in the soil. usually a stem or a leaf. After few days, the cut part will form adventitious roots and begins to grow as a new plant. This method is very useful in rose, hibiscus, bougainvillea, etc. Layering. In this, the stem of the plant is bent to the ground and covered with soil. Adventitious roots emerges from the plant parts, whereas it is covered with soil. Example for layering is Nerium plants. Grafting. Two plants are joined together in such a way that two stems joined and grow a single plant. Both plants are tied up with a plastic cover. One plant which roots are present is called stalk, and without roots is called scion. Grafting method is very useful in many plants such as mango, apple, lemon, etc. Conclusion. This method of artificial vegetative propagation is favored for the production of a large number of plants at once. Sexual reproduction. Reproduction is necessary life process for continuation of life by producing offsprings. The mode of reproduction is asexual reproduction and sexual reproduction. In sexual reproduction, male organisms produces male gametes and female organisms produces female gametes. Male gametes and female gametes fusions together to form zygote. This zygote further develops into new organism. Male gametes, that is sperms, and female gametes, egg cells. The fusion of these sperm cell and egg cell is called fertilization. Fertilization is two types: external fertilization, internal fertilization. The fertilization occurs outside the body is called external fertilization. Most of the aquatic animals such as frog, fishes are seen external fertilization. The fertilization occurs inside the body is called internal fertilization. The terrestrial animals such as human beings are example for internal fertilization. Reproduction in human beings. Let us observe male reproductive system. In male reproductive system, a pair of testes, a pair of vas efferentia, a pair of epididymis, a pair of vas deferentia, a pair of seminal vesicles, ejaculatory duct, penis, and urethra are seen. Some of the glands such as prostate and Cowper's glands also present in male reproductive system. Testis. Testis is located outside the body in a pouch is called scrotum. In each scrotum, there are highly coiled tubules are present. These tubules called seminiferous tubules. These seminiferous tubules produces sperm cells. And also testosterone hormone also secretes here. Vas efferentia epididymis. Vas efferentia. Seminiferous tubules enters into vas efferentia. Vas efferentia carries sperm cells into epididymis. Epididymis. Sperm cells temporarily stored in this place. Vas differentia. From each epididymis arises as vas differentia. Seminal vesicles. Seminal vesicles opens into vas differentia. In this seminal vesicles, seminal fluid is produced. Seminal vesicles and vas differentia together forms ejaculatory duct. In behind the ejaculatory duct, we will find two glands. They are prostate gland and Cowper's gland. These two glands secrete some secretions which are enters into ejaculatory duct. Urethra. In male reproductive system, urethra is the pathway of urine and sperm cells. Journey of sperm cell. Sperm cells produces at seminiferous tubules and enters into epididymis, vas efferentia, ejaculatory duct, and through urethra the sperm cell eliminates from the body. Structure of sperm cell. In sperm cell we find acrosome, head with nucleus, neck piece with a mitochondria and a tail. Cowper's glands, prostate gland and seminal vesicles produces some secretions. All are together called seminal fluid. Seminal fluid and sperm cell both are together called as semen. Female reproductive system. In female reproductive system, a pair of ovaries, a pair of oviduct, that is fallopian tubes, uterus, cervix and vagina are present. Ovaries. A pair of ovaries located in the abdominal cavity near the kidneys. Ovaries produces egg cells, that is ovum. The ovary matures one egg cell at a time in every alternate month. And also it secretes female sex hormones like estrogen and progesterone. Oviduct. A pair of oviduct or fallopian tubes arises from uterus. It is the site of fertilization. Uterus. Uterus is also called womb, where the implantation and development of baby takes place. Cervix. The uterus opens into vagina through the cervix. Fertilization. The fusion of egg cells and sperm cells is called fertilization. After fertilization, the zygote forms. Further, zygote develops into embryo. After maturing the ovum, it releases from ovary. The release of the egg from the ovary is called ovulation. After ovulation, the egg enters into widened funnel shaped fallopian tube. The egg passes through fallopian tube to meet sperm cells. Development of the embryo. After releasing the egg into fallopian tube, it started journey to meet sperm cells. After fertilization, the fertilized cells move towards the uterus and finally it settles down in the uterus. The process of reaching the embryo into the uterus is called implantation. Membranes around the embryo. Chorion. During development of embryo, tiny finger-like projections grow from the outer side of the uterus is called chorion. Tissue of chorion and the adjacent part of the embryo tissue makes another layer called placenta. Placenta is formed after 12 weeks of pregnancy and become an important structure for the nourishment of the embryo. There is no direct flow of blood to mother to embryo. The placenta membrane allows the material by diffusion, mainly carbon dioxide, oxygen, nutrients, and waste materials. The another layer is amnion. Amnion grows around the embryo by itself. The cavity around the amnion filled with a fluid called amniotic fluid. The embryo develops in this fluid and protected. Umbilical cord. It contains a major blood vessel that connect to placenta to embryo. From the third month of pregnancy, the embryo is called fetus. The fetus developed fully average in 9 months of period. This period is called gestation period. Parts of a flower. A flower is the reproductive part of the plant. A flower contains four parts. They are sepals, petals, stamens, and pistil. Sepals. Sepals are outermost parts. These sepals protect the flower before it blooms. Petals. Petals are either white or in colored. The petals attract the insects to pollination. Sepals and petals do not take part directly in reproduction. Stamens. Stamens is a male reproductive part of a flower. Stamens contains anther and filament. Anther produces male gametes, that is pollen grains. Pistil. It is the female part of the flower. Pistil contains stigma, style, and ovary. In this ovary contains egg cells or ovule. These egg cells are the female gametes. Male reproductive parts in a flower. Filament and anther, together called stamens. Anther produces pollen grains. These pollen grains are male gametes in a flower. Female reproductive parts in a flower. Stigma, style, ovary, together called pistil. Ovary produces egg cells or ovule. These are female gametes. According to sexual parts in a flower, flowers are two types: unisexual flowers and bisexual flowers. A flower contains stamens and pistil. Both are together in a flower. Is called bisexual flower. Example: rose, hibiscus, etc. The flower which contains either stamens or pistil are known as unisexual flower. Example: papaya, watermelon, etc. If a flower contains only stamens, is called a male flower. Or if a flower contains only pistil, is called female flower. These are unisexual flowers. Pollination. The transfer of pollen grains to the stigma is called pollination. Pollination is two types: self-pollination and cross-pollination. When the pollen grains of a flower reaches to the stigma of the same flower is called self-pollination. If the pollen grains of a flower reaches to the stigma of another flower of the same plant is called cross-pollination. Cell is the structural and functional unit of life. Cell contains many organelles. In between them, nucleus is play an important role in cell division. Nucleus contains chromosomes. These chromosomes are responsible for genetical material. Cell division is a controlled process. Cell division is an important process for all organisms. Cell division means cell dividing into two daughter cells. What is the use of cell division? Cell division helps in growth and body development. Our life starts with a single cell called zygote. After many cell divisions, our body develops. There are two phases in cell cycle. They are interphase and division phase. Before cell division, cell undergoes many changes. There are four stages in interphase. They are G1 phase, S phase and G2 phase. In G1 phase, the cell size grows, and in S phase, the cell synthesizes a complete copy of the DNA in the nucleus. In G2 phase, the number of cell organelles were increases. The duration of the cell cycle total 24 hours. The G1 phase might last about 11 hours and S phase about 8 hours, G2 phase about 4 hours and finally cell division phase about 1 hour. Completion of all changes in the cell is ready to divide. After the cell division, cell undergoes into interphase, that is rest phase. Cell division. Our body is made up of 15 trillions of cells. These cells are two types, somatic cells and germ cells. Various parts of our body is divided from these cells. These organs are derived from somatic cells. Reproductive organs such as ovaries and testes are developed from germ cells. The cell divisions are two types, mitosis and meiosis. Mitosis. In mitosis, the mother cell produces two daughter cells which are mostly resemble to mother cell. In mitosis, there are some phases: prophase, metaphase, anaphase and telophase. Cell did not divide directly. It has some sequences. In cell division, first the nucleus divides and then next cytoplasm divides. The division of nucleus is called karyokinesis and the division of cytoplasm is called cytokinesis. Prophase. In prophase, the nuclear membrane will disappear. The chromosomes contrast and spiral. It is visible under the microscope. The chromosomes split into two chromatids. These chromatids are attached at centromere. Centrioles will reappear. Each from centriole, fine thread-like structures are appears. These fibers are called spindle fibers. By the end of prophase, nuclear membrane will disappear. Chromosomes contrast. Chromosomes split into chromatids. Centrioles will reappear. Spindle fibers forms. Metaphase. Chromosomes now move to the center of the cell and arrange on the plate, that is equatorial plate. Spindle fibers are attached to the centromere of each chromosome. Anaphase. At this stage, spindle fibers pull the chromosomes to opposite ends. As a result, half number of chromosomes move to each of the two opposite poles. Telophase. By the beginning of this phase, the chromosomes complete the movement and will be at each end of the cell. By the end of telophase, cell will have two nuclei, one on each side. Spindle fibers now disappears. Cytokinesis. A new membrane is formed at the equatorial plate during this phase. This divides the cell into two daughter cells. Thus, at the end of mitosis, two daughter cells which are exactly similar to the mother cell are formed. Conclusion. By the end of mitosis, the mother cell produces two identical daughter cells. Before cell division, the chromosomes double in number and distributed equally to the daughter cells.